The idea of freezing a human body to preserve it for future revival has long captured the human imagination, blending science fiction with the tantalizing promise of immortality. Cryonics, the practice of cooling bodies to ultra-low temperatures in hopes of resurrecting them when medical technology advances, has sparked both fascination and skepticism. From the frozen remains of Ted Williams to the hundreds of patients preserved in cryonic facilities, the question persists: can cryogenics truly bring you back to life? In this video, we will explore delves into the science, technology, ethics, and challenges of cryonics, separating fact from speculation to uncover the truth behind frozen bodies. What is cryonics? Cryonics is the process of preserving a human body, or sometimes just the brain, at extremely low temperatures, typically using liquid nitrogen, with the goal of reviving the individual in the future. Unlike cryobiology, which studies the effects of low temperatures on living organisms, cryonics is speculative, aiming to preserve biological structures until medical advancements can reverse death and restore health. The process is initiated shortly after legal death, when the heart stops, but before irreversible cellular damage sets in. Cryonic preservation involves several steps: cooling the body to halt decay, replacing blood with cryoprotectants to prevent ice crystal formation, and storing the body in a cryogenic chamber at approximately minus 196 degrees Celsius, minus 321 degrees Fahrenheit. Organizations like the Alcor Life Extension Foundation and the Cryonics Institute, both based in the United States, have been offering these services since the 1970s, with over 500 individuals currently preserved and thousands more signed up for future cryopreservation. The history of cryonics. The concept of cryonics emerged in the 1960s, rooted in the optimism of scientific progress. In 1962, physicist Robert Ettinger published The Prospect of Immortality, a book that laid the foundation for cryonics by proposing that freezing bodies could preserve them until future technologies could revive and cure them. Ettinger founded the Cryonics Institute in 1976, and the first human cryopreservation occurred in 1967 with James Bedford, whose body remains preserved at Alcor. Cryonics gained public attention through high-profile cases, such as the 2002 preservation of baseball legend Ted Williams, which sparked legal disputes and media frenzy. Despite its niche status, cryonics has grown into a small but dedicated movement, fueled by advances in biotechnology and a cultural fascination with defying death. However, the practice remains controversial, viewed by many scientists as pseudoscience due to the lack of evidence for successful revival. The science of cryopreservation. To understand whether cryonics can bring someone back to life, we must examine the scientific principles behind cryopreservation and the challenges of reviving a frozen body. The cryopreservation process. Cryopreservation begins immediately after legal death to minimize cellular damage. First, the body is cooled in an ice bath to slow metabolic processes. Blood circulation and respiration are artificially maintained to deliver oxygen to tissues. Next, cryoprotectants, chemicals like glycerol or dimethyl sulfoxide, are introduced to replace bodily fluids, reducing the formation of ice crystals that could damage cells. The body is then cooled to minus 196 degrees Celsius in a controlled process called vitrification, which transforms tissues into a glass-like state without freezing. The body is stored in a dewar, a vacuum-insulated container, filled with liquid nitrogen, where it can theoretically remain preserved indefinitely. For those opting for neurosuspension, only the brain or head is preserved, based on the belief that future technologies could regenerate a body or upload consciousness to a digital substrate. Cellular and tissue challenges. The primary obstacle to cryonics is cellular damage. Even with cryoprotectants, some ice crystal formation or chemical toxicity can occur, particularly in complex organs like the brain. Freezing and thawing processes can cause cell membranes to rupture, proteins to denature, and synapses to degrade. A 2015 study in cryobiology demonstrated that vitrification can preserve brain tissue in small animals, such as rabbits, with minimal damage. But scaling this to human brains remains untested. Reviving a cryopreserved body would require repairing this damage at a molecular level. Nanotechnology, often cited by cryonics advocates, could theoretically repair cells, but such technology is decades, if not centuries, away. Additionally, the brain's delicate neural networks, which store memories and personality, must remain intact for a revival to preserve the individual's identity, a feat that current science cannot guarantee. Defining death. Cryonics hinges on the definition of death. Legal death occurs when the heart stops, but biological death, irreversible cellular breakdown, takes longer. Cryonicists argue that individuals preserved immediately after legal death are not truly dead if their cellular structures remain viable. However, critics note that current medical technology cannot reverse clinical death, and the brain begins to suffer irreversible damage within minutes of oxygen deprivation. A 2018 study in Nature showed that pig brains could be partially revived hours after death, but this involved restoring cellular function, not consciousness, highlighting the gap between preservation and true revival. The technological hurdles. Reviving a cryopreserved body requires advances in multiple fields, none of which are currently feasible. Repairing cellular damage. To revive a frozen body, future technology would need to repair billions of cells damaged by cryoprotectants, ice crystals, or ischemia, lack of blood flow. Molecular nanotechnology, proposed by figures like Eric Drexler, could theoretically rebuild cellular structures, but practical applications remain speculative. Current research focuses on nanoscale drug delivery, not tissue reconstruction, regenerating organs. If the body is too damaged, revival might involve growing new organs or an entire body. Stem cell research and 3D bioprinting have made strides. Researchers grew functional heart tissue in a lab in 2020, but creating a fully functional human body is far beyond current capabilities. For neurosuspension patients, transferring consciousness to a new body or digital platform raises philosophical questions about identity and continuity. Reversing aging and disease. Most cryonics patients are preserved due to terminal illnesses or aging. Revival would require curing the original cause of death, such as cancer or Alzheimer's. While gene therapies and immunotherapies show promise, for example, CAR-T-cell therapy for leukemia, curing all diseases remains a distant goal. Aging itself, driven by complex processes like telomere shortening and cellular senescence, is only partially understood, with no comprehensive cure in sight. Uploading consciousness. Some cryonicists propose that future revival could involve uploading consciousness to a digital substrate, bypassing biological revival. This assumes that consciousness can be fully mapped and transferred, a concept explored in projects like the Human Connectome project. However, a 2023 study in Neuron estimated that mapping a single human brain's connectome would require petabytes of data and decades of processing, with no guarantee of preserving subjective experience. Ethical and philosophical questions. Cryonics raises profound ethical and philosophical issues. Is it ethical to offer a service with no proven success? Cryonics facilities charge $80,000, $200,000 for preservation, often funded through life insurance. Yet there's no evidence that revival is possible. Critics argue this exploits hope, while proponents view it as a rational bet on future technology. The question of identity is central. If a revived person's memories or personality are altered, are they still the same individual? Philosophers like Derek Parfit argue that identity is tied to psychological continuity, which cryonics may not preserve. Additionally, reviving thousands of cryopreserved individuals could strain future resources, raising questions about who gets priority and how they integrate into a radically different society. Consent is another concern. Some patients, like children or those preserved posthumously, may not have fully consented. The case of Ted Williams highlighted family disputes over cryopreservation, underscoring the need for clear legal frameworks. The current state of cryonics. As of 2025, cryonics remains a niche field with no successful revivals. Alcor and the Cryonics Institute store approximately 500 bodies and heads, with thousands more registered for future preservation. Advances in cryobiology, such as the 2016 vitrification of a rabbit brain by 21st century medicine, show progress in preserving neural structures, but these are far from human applications. Public perception of cryonics is mixed. A 2021 Pew Research survey found that 31% of Americans believe cryonics could become viable by 2100, but 62% view it as unnatural or unethical. High-profile advocates like tech entrepreneurs Peter Thiel and Elon Musk have expressed interest. But mainstream science remains skeptical, with the American Medical Association calling cryonics unproven in a 2020 statement. Real-world analogies and lessons. Cryonics draws parallels to other speculative technologies. Organ transplantation, once considered impossible, became routine by the late 20th century. Similarly, advances in regenerative medicine could make cryonics feasible, though the timeline is uncertain. Historical attempts at preservation, like ancient Egyptian mummification, reflect humanity's enduring quest to defy death. But unlike mummification, cryonics aims for reversibility. The case of Ötzi the Iceman, a 5,300-year-old mummy discovered in 1991, shows that biological material can be preserved for millennia, but Ötzi's tissues were not viable for revival. Cryonics faces a similar challenge. Preservation is possible, but restoration is not. Conclusion. Cryonics embodies humanity's audacious dream to conquer death, but it remains a speculative endeavor with no proven path to revival. The science of cryopreservation has made strides, preserving tissues with unprecedented precision, but reviving a human body or brain requires breakthroughs far beyond current capabilities. Ethical concerns, from consent to societal impact, further complicate the picture. While cryonics offers hope to those who fear oblivion, it also demands a leap of faith in a future that may not materialize. For now, the frozen bodies in cryonic dewars remain silent, waiting for a science that may one day answer whether they can truly be brought back to life.